Good evening, everybody. It's Doc Kyle, and tonight we're going to talk about nucleic acid testing. So, there are other ways to do nucleic acid testing apart from the things that we will discuss tonight, but I will talk about the most timely na mga tests, no? Like your PCR. So, we also have your northern blot, and then western blot, southern blot, which tests for different types of biomolecules. But we're just gonna focus on PCR tonight. So first is, what is cloning? So cloning, in the context of, I think this will be a better presentation. So we have what we call your bacteriophage. This bacteriophage, they are viruses that attack or attach to your bacteria. Thus the name bacteriophage. So what will happen is this bacteriophage will insert their DNA into the DNA of bacteria. This is a bacteria over here. They will insert their DNA into the bacteria. And after they inserted their DNA within the bacteria, as the bacteria undergoes binary fission, the DNA of the bacteriophage hijacks the DNA of the bacteria, making it produce more bacteriophage DNA and viral molecules. Eventually, the bacteria is now filled up with lots more of bacteriophages. Then it will eventually pop, and then it will liberate more bacteriophages, thus continuing the cycle. So in the context of cloning, if we have what will happen is we will combine bacteriophage DNA and some human DNA. So after that, after we combine them, that's what we call recombinant DNA. So what's the point? No, what's the point of combining this? So remember that binary fission, especially in bacteria, is way faster than mitosis in eukaryotes. No, so if you want to get numerous copies of a gene, no, numerous copies of a gene, then we if we're gonna wait for a example, if we harvest the gene of a cow, it's gonna take so long. So what we will do is we'll take advantage of the rapid division of the bacteria to harvest this protein, this DNA copies, no. So in order for you in order for us to insert our DNA into rapidly populating bacteria, we first need to get a vector. No, this is your vector, the phage. So that is our viral vector. So first we combine our DNA with phage DNA. We mix them and join them. And then we add our solution of katong phage bacteriophages in a plaque on lawn of bacteria. So here you can see it's filled with bacteria. And then we let loose our recombinant DNA containing bacteriophages. And then eventually they will reproduce, the bacteria will reproduce. And then what's all that's left are your bacteriophages, because like I said, your bacteriophages will hijack this bacteria, and then eventually it will pop, making more bacteriophages. So remember, gisakyan lang sa bacteriophage ang mga bacteria na nag-undergo og binary fission, and then eventually the bacteria that will undergo binary fission will eventually pop, and then it will release lots of new bacteriophages with our recombinant DNA. No? So think of it, ha? Gamay, if you're just gonna add one human plus one phage DNA, and then you go through this process, you're gonna be left with lots and lots of bacteriophages with recombinant DNA. So that's quite bonkers if you ask me, no? So a clone is a genetically identical population of organisms. So it's the basis of genetical identity. No, it's basically a copy. No, so like I said earlier, it is the introduction of a section of DNA into a genome which it can be reproduced many times. So kato. And then the vector is a carrier molecule for transfer of genes in DNA recombination. So DNA recombination simply means that you add foreign DNA to another DNA. So what will come out is a completely unique DNA strand. So a plasmid is a small circular DNA molecule. It usually contains antibiotic resistance and is used often for cloning. So how bacteria develops antibiotic resistance is that, so naman like in humans, there are people who are really inherently resistant to infections. So that is the opposite in bacteria. In bacteria, there are bacteria that are inherently, they are the same species, same type of bacteria, but there are minute differences between them that makes like a certain population of bacteria resistant to antibiotics. So when you give antibiotics to the certain types of bacteria, then this two will die and this one will continue to live. And then this bacteria will now binary fission, managhan lang gyapon na siya. So we have what we call horizontal transfer. No? So sometimes there is what we call pili, and this pili is like a tube, and then example silingan, this bacteria over here doesn't have antibiotic resistance. Sometimes this guy that has antibiotic resistance will share some of its plasmid with this guy, so that this one also will have antibiotic resistance. Of course, all the offspring of this bacteria, after it has undergone binary fission, will have antibiotic resistance. So plasmids is more of kanang horizontal, no? Plasmid, I want you to understand is usually your bacteria, your prokaryotes usually have one circular type of DNA. That's all they have. But there are non-main DNA na mga non-main kanang genetic material. So this mga genetic material, they don't encode for structural and kanang structural proteins or functions that will make the bacteria survive and live. They just contain specific na mga actions for encoding, like I said earlier, antibiotic resistance. That's why they can be shared, no? Okay, if share nimo imuhang structural and functional na DNA, matay of course bacteria, no? So going back here, so even though it's quite rapid, no, it's quite fast, and it produces a lot of genetic material. The problem is, it's still quite hago because you need to have bacteria plaque pa, you need to harvest your bacteriophages pa and such. So it is possible to increase the amount of DNA, no, without cloning. So we have what we call this PCR, polymerase chain reaction. So PCR copies both complementary strands of the desired DNA sequence. So nasikat na yung PCR because of the advent of COVID. Remember that it's really hard to detect viruses. We can only, for the most part, whenever, if you ever had a viral infection in your life, most probably we didn't detect the virus directly. We used like dengue, we used check antibodies if you developed against kanang mga viruses, or sometimes we just use our clinical eye. Especially sometimes when you had the flu, you never had a flu test, you just had a CBC. and the CBC is just a clue on what infection you have. So, what happens with PCR, no, in the context of viruses, is remember that your viruses are nothing more than some protein and genetic material, either RNA or DNA. So, and remember that for the most part, your bacteria are intracellular parasites, no? Meaning, in order for us to culture them, to make them, to propagate them, no, to populate them rather, and then to test them to know what bacteria they are, to know what virus they are. The culture media that we need is basically cells, no. So usually we use HeLa cells, no. But I digress. That's both very tedious and it's very hard to do. I have never experienced it to culture viruses. So the best thing, no, the gold standard, no, is what we call your nucleotide amplification test. Meaning, we're gonna amplify and look for your genetic material in your samples, from your nose, from your, to check if there are active viruses or there is viral replication in your nasal swab, no, or in your blood or in your poop. And that's what we do with PCR. So, this polymerase chain reaction, what it does is it amplifies, no, it amplifies and it doubles your genetic strain. So what I want you to understand here is this is a table. So this is the target sequence, meaning this is the DNA of your bacteria, your virus, no? Example, your SARS-CoV-2 virus. No? This is the sequence. No? This is the specific genetic sequence na makita nato sa SARS-CoV-2. If it is present in your nasal swab, then we have what we call your primers. They will attach. This is a primer here, and this. So your primers are specific. They are specific, meaning this is the target, ano man, no, DNA. This is target RNA in the context of ano. So reverse transcriptase, I think it's gonna be tackled more in your clinical chemistry or microbiology in your later years. But for now, we're just gonna talk about in this context, no. So your primers are specific na strand of complementary DNA, no. So what we will do, so first we should know what DNA. So what DNA does the virus have, no? What specific DNA strand, no? And what strand is specific to that virus, no? That's why it was it took a long time before we could make PCR readily available to the entire world, because viral mapping of SARS-CoV-2 was still being done. So how can we detect something if we don't know what it is, no? So after the viral mapping was done, genetic mapping was done, the companies who make PCR then made their primers. Primers are relatively short chain na mga DNA strands, complementary DNA strands that will bind to a specific part, no? So it's usually countercurrent, no? Usually it's the downstream and the upstream, no? 5 prime, 3 prime. Anyway, it binds to a specific spot, no, of the target genetic sequence. And then a DNA polymerase will act on that primer and it will go up, example upstream, it will go up and it will fill in the missing, it will fill in the missing nucleotides, no? So this is your primer over here, and then your polymerase will notice the primer, and then it will continue, it will start to make the complementary strands of this target. So this is the target DNA strand over here. Kani siya, kaning sa baba. This is your primer, and then this is your DNA polymerase, and it will fill it up. Okay? So, so kana siya. So that's the first step. So kani siya. So the first so if we're gonna talk about DNA, it's a double strand. So before we can before the primers can attach, no? Before the primers can attach, it needs first to unwind to separate. So in order for them to separate, we need to introduce high heat, 95 degrees, no? So that's what we call denaturation. So your DNA is denatured and it's split apart. Next step is annealing. It is when your primers attach. And then extension is when your DNA polymerase works. So if it is that simple, why didn't we develop it way before? Why isn't this widely used? No? Especially in the context of viral infections. Number one, it's just been recently that a virus really wreaked havoc on the world, no? Wreaked havoc on the world. So, wala kayo siya big deal because most of the viruses, even COVID, SARS-CoV-2 is quite self-limiting. Ito. That's it. Then number 3, it's really hard to find a DNA polymerase, no? That can handle 95 degrees Celsius. So that DNA polymerase, remember, is a protein. And as we all know, proteins tend to denature when exposed to high heat. So, it's really hard to find a resilient DNA polymerase. Good thing that we have this what we call your Taq DNA polymerase, which is your Thermus aquaticus. No? So your Thermus aquaticus is extracted, is where your heat-resistant polymerase is extracted. No? So your Thermus aquaticus live here, no? In thermal vents, no? Hydrothermal vents. So it is believed that your it is believed that life actually started here in hydrothermal vents, no? So it's one of your what we call archaeobacteria. It's really old, no? So kato, and it lives in very hot places, no? So that's why its DNA polymerase is quite resilient to heat. Okay, going back. So after na to elongation, no? So okay, repeat. First exposed to high heat, matunga siya. That's your denaturation. Your primers then attach. That's your annealing. Annealing over here. And then your DNA polymerases works, and that's now your elongation. Then that is one cycle. Then after there is full elongation na of the target sequence, then we will expose it again to 95 degrees Celsius, your heat, so that it will now again denature and split, and then your primers will again attach, and then your polymerase will work. So what happens is there will be an exponential growth, no, of your sequences, no. So every cycle your DNA will double, no. So exponential, it's no exponential. So padaghan siya ng padaghan. So the beauty of kaning PCR is even if small lang ang amount sa DNA, we can still detect it, no. So what happens is after we get a lot of copies na, we can check if our primers, no, sometimes we add ano man good in your real-time reverse transcriptase PCR. We have what we call your quenchers and your fluorophores, so we can check if there is kanang increases in the target gene amplification. So what we call is amplification, meaning amplification, padaghan ng padaghan ang genes, no. So we can check if there is increase in amplification because there are, like I said, mga mga fluorophores, no, that we can detect, no. So if there is an increase in the total gene the total target gene concentration, meaning most probably you have that virus, you have an actively dividing virus or some viral particles in your nose. So that's why you are tested PCR positive. Okay? So that's the basic gist of PCR. No? So the most important part of the science of behind PCR is the primers. No? So it needs to be long enough so that it will binds to that specific start of that target gene, no? And it should be short enough para dili siya ato kamahal. Mind you, this is very expensive to develop, no? So, before ta makahimo og primer, gibuhat sa gi-cloning sa na siya, gi-recombinant DNA sa na siya, no? So, I'm not saying that PCR has superseded cloning because still cloning still has kanang katong bacteriophage, it still has a part, no, especially in the prototyping, no, or kind of initial stages of development for reagents for PCR, no. So they usually have 18 to 30 bases, and they also must have optimal binding properties such as amount of GNC to allow them to anneal. In addition, two primers should contain similar amounts of GNC so that they have the same. So that is all, no. So hopefully in the future I will be able to lecture you. I will be able to talk to you about kanang PCR in the context of kanang RT-PCR, reverse transcriptase, no? So PCR na basic na conventional real-time PCR is used when the target molecule is DNA, no? So we also have another term here, reverse transcriptase, no? So your reverse transcriptase PCR is a type of PCR that handles or detects RNA-based na mga molecule. So your reverse transcriptase is an enzyme that converts your RNA into DNA, no? So it's usually found in your HIV/AIDS, no? No, mga virus, your HIV/AIDS tuloy na. Yeah, your HIV na virus, no? Your HTLVs, no? So they are essentially RNA viruses, but they make their own DNA to incorporate to the host cell. So that's a topic for another day. So if you have any questions, feel free to PM me. So good night, guys. God bless.